This is podcast number 5. So in this podcast, we will learn about the structure of DNA, showing the double helix and base pairings, what goes with what when you're drawing a DNA, and that is a skill that you're going to use over and over in this class, also in lab. And then the idea of semi-conservative DNA replication, how one DNA molecule becomes two DNA molecules. We'll talk about that. So this is one way you can draw a DNA molecule. Here are the 3 phosphate groups, 1, 2, 3. Here is the sugar that is the middle of the DNA molecule, and then this thing here is called the base. And there's different kinds of bases: adenine, guanine, cytosine, and thymine. But the sugar is the same, and the phosphates are the same for all of them. The sugar is different for DNA versus RNA. DNA has a missing oxygen on this sugar, which is not there in, or which is there in RNA's sugar. So DNA is called deoxyribonucleic acid because it's mixing that oxygen, deoxy, versus ribonucleic acid over here. Okay, RNA, this base is an A, so this is called A, TP. T means tri, 3 phosphates, and then this is DATP, the DNA version of that base. Okay, and if you rotate this structure 90 degrees, or if you draw it in a different direction, here's those 3 phosphates, here's the sugar, and here's the adenine base. Again, this part is different between A, T, C, and G, adenine, thymine, cytosine, and guanine. This part is going to be the same in all DNA molecules. This part is going to be the same in all DNA and RNA molecules. This carbon right here is the one that's missing the extra oxygen in DNA, and that's why it makes it deoxyribonucleic acid. If this was an RNA sugar, it would have an oxygen, just like there's an oxygen over here. So one thing that's really important in looking at the structure of DNA is the numbering of these carbons on the sugar. So this is number 1, number 2, number 3, number 4, and number 5. These are the carbons on the sugar. Again, number 1, number 2, number 3, number 4, number 5. Now, for the chemistry of this is called 1'. That little hash mark is is pronounced prime. 1', 2', 3', 4', 5'. The reason why this is important is because these nucleotides, that's what this whole thing is, get connected to each other, and 5' is always the beginning of a chain, and then you add on another one of this whole thing to the 3' end of this sugar. So if I was going to add another A, I would add it right here, and it would start 5' here. So if I'm looking at a chain of nucleic acid, always, 5' is the number 1 nucleotide, and 3' is the end of the chain. And the chain can only get bigger by adding on to the 3' end. You can't add on to the 5' end. So again, 5' is always the beginning. You add on new ones to the 3' end. Wherever it ends, there'll be a 3' end hanging off. So this is always the end. That's always the beginning. Okay. Nice big diagram here to show you again. 1, 2, 3 phosphates, sugar in the middle. I know that this is DNA, DATP, because it's missing this oxygen here on the 2' carbon. And then this happens to be an A, base because it has adenine for the base. If it was a thymine over here, that would be called T. If it was a guanine over here, it would be called G. If it was a cytosine over here, it would be called C. Okay? And again, the 5' end is always the beginning of the chain. You can't add on to this 5' end. You can't. This whole thing here is called a nucleotide. And if I want to add another nucleotide to this, let's say I wanted to add a T to this A, I would add it on at this 3' end. Okay? So the 5' end is always the beginning, and the 3' end is the end. And the chain can be 1,000 base pair, 1,000 nucleotides long, 10,000 nucleotides long, 50,000 nucleotides long. Number 1 would always be have a free 5' end, and number 50,232 would always have a free 3' end. And if I wanted to add another one to that 50,232 to make it 50,233, I would add it on to this 3' end. Okay, so when you're putting it into a DNA molecule, this is what we just looked at. So the sugar is in the middle, the phosphate group's hanging off the side. Here's the phosphate, here's the sugar in the middle, and then here's the base that they're going to say in this next diagram, okay? And this phosphate attaches to the next sugar, this phosphate attaches to the next sugar, this phosphate attaches to the next, and the bases are facing each other. So you have a chain of nucleotides going down one side and another chain going down the other side, and they're connected to each other by the bases. When you put them in a big long chain, they curve past each other in what's called a double helix, okay? And then these rungs of the ladder are the bases in green here, connected together by hydrogen bonds, which we'll talk about in a second. So Watson and Crick were the guys who first described this model for DNA. There had been some other models described for the structure of DNA before that. Theirs was the first one that ended up being correct. But even in their first paper where they described this model, they made a mistake, and that is that they didn't recognize that the two strands are anti-parallel. What that means is if this is the 5' end of this strain, and this is the 3' end of that strain of DNA, then the other chain that is attached to it is in the opposite orientation. So that the 5' end is here and the 3' end is there. So they didn't figure that out, and if they had figured that out, this phosphate wouldn't be here, it would be up here. So anti-parallel is what that's called. 5' to 3' in one direction, and the chain on that it's attached to in the other direction is going the opposite way. 5' to 3', top to bottom. This is 5' to 3', bottom to top. Okay, so I mentioned a hydrogen bond, and that is one of the kinds of chemical bonds that you find in biological systems. There's a couple other ones that we'll talk about. This hydrogen bond is a bond where there's a partial charge on an atom. So this is an oxygen. That little squiggly symbol there is a Greek lowercase delta, and it means partial. So oxygen is a big atom. It has electrons buzzing around it, and the electrons tend to spend more time on one part of the molecule than on another part of the molecule. And that makes one part of the molecule partially negative because the electrons tend to hang out there more often, and another part partially positive. Hydrogen doesn't have electrons hanging around it often, and so it'll be partially positive. and this partial negative and partial positive will tend to attract each other. This is usually oxygen bound to another molecule will have a partial negative or partial positive charge. For example, oxygen in water, H2O, oxygen is very negative and the hydrogens are very positive. So it's not that you have one hydrogen on one side, one hydrogen on the other side, and the oxygen in the middle. The hydrogens tend to be stacked up on one side of the oxygen atom, making one part of the water molecule partially negative and one part partially positive. And so they can form these partial charges, and water molecules stick to each other by these partial charges, which are called hydrogen bonds. So hydrogen bonds are weak interactions. They're typically between oxygen atoms or nitrogen atoms and hydrogen. And they cover a distance of 2 angstroms. Angstroms is a very small unit of length. Like a meter, you have some idea that that's like about as big as a yardstick. An angstrom is much, much, much smaller than that. And it's just a unit of length. And if things are 2 angstroms apart, they can be held together by hydrogen bonds. But if they are more than 2 angstroms apart, that's too far for a hydrogen bond to hold them together. An ionic bond is a fully negative charge interacting with a fully positive charge. So this little delta here means that this is a partial negative and a partial positive. Partial negative meaning that in a molecule, the electrons like to hang out in one part of the molecule more than they like to hang out in another part of the molecule, but they're still zipping around the whole molecule. And so one part of the molecule is partially negative, one part of the molecule is partially positive. In an ion, they have an extra electron, a full, at least one full extra electron, or they're missing an electron. And that gives them a net positive charge if they're missing an electron, as opposed to how many protons they have. If the number of protons and electrons are not balanced and they're missing electrons, they'll have a positive charge. If they have more electrons than they have protons, then they'll have a negative charge. And if you have a full negative charge and a full positive charge, they can be attracted to each other, sort of like a magnet. And that's called an ionic bond. And an ionic bond is stronger than hydrogen bond. And because it's stronger, it can hold things up to 4 angstroms apart from each other. So twice as far as a hydrogen bond. And a covalent bond is an even stronger bond. And that's when 2 molecules are so close together that the electrons actually orbit orbit all the atoms in the molecule. So the bond between 2 separate things, like this nitrogen here and this carbon here, the electrons would be zipping around both of those atoms. And because they're sharing electrons, it tends to bring those 2 things closer together. And that's a very strong bond, and that's called a covalent bond. So of these 3 bonds that we'll talk about, hydrogen bonds are the weakest. Ionic bonds are in between. Covalent bonds are the strongest. So these are the weakest. They are the easiest to break. These are the hardest to break. It takes the most energy to break a covalent bond. It takes the least energy to break a hydrogen bond. Okay. So Watson and Crick were trying to figure out the structure of DNA. They can't use just like a microscope to go look at DNA. It's too small a molecule. So they use some other techniques. One of the techniques is called X-ray diffraction. And in X-ray diffraction, if you have a large pure sample of a substance like DNA, for this example, you can bounce X-rays off of that large pure sample. And the angle in which the X-rays come off your sample, tells you something about how the atoms are arranged in that substance that you're trying to see. And the point of this was that this technique was really important to figuring out the structure of the DNA. So one of the things they were wondering about was, where are the bases? Are they on the inside like this, or are they on the outside? Other molecules, models had them on the outside. Where are the phosphates? That sort of thing. By using X-ray diffraction, they're bouncing X-rays off of these individual molecules and then figuring out the angles from those. They can get an idea of where stuff is. Okay, they used some data from an X-ray diffraction pattern of a colleague of theirs in the same academic department as theirs. And that colleague was Rosalind Franklin. She's a woman working in the early 50s in this very conservative department, and she wasn't taken very seriously. But she had the best data. They used her images from her data to come up with their model. And just by contrast, there was another author that published in the same journal as Watson and Crick when they're describing the structure of DNA. And he also published his X-ray crystallography data. But you can see that the quality of these images is not as sharp as the quality of these images. So for example, you can see this pattern here, and you can't really see the third band here, or as much detail around the center point here. Now, I'm not used to looking at X-ray crystallography. I don't imagine you are either, but to a trained person, this versus this would make a big difference. So they said that, they looked at other people's data. They didn't give them credit for it in the paper, and those people were Rosalind Franklin. They never gave her credit and badmouthed her for a long time. She didn't get the credit she deserved, and unfortunately she died early. But now we recognize that her contribution to the structure of DNA was critical, and that it's likely that Watson and Crick wouldn't have been able to figure out what they figured out without her data. And now, posthumously, after she's died, she's starting to get credit. She was, her name was put on a recent Mars probe, and she's recognized for making a big contribution to science early. But unfortunately, women have gotten a really bad treatment in science, not getting the credit they deserve for a lot of important discoveries like this one. Okay. So, part of the big mistake, or part of the initial structure that they proposed, indicated that certain bases interacted with each other with a hydrogen bond. For example, guanine and cytosine. They didn't, in their original paper, they didn't have 3 hydrogen bonds between guanine and cytosine, but indeed there are 3 hydrogen bonds between guanine and cytosine. All 3 of these atoms are within 2 angstroms, and indeed there are 3 bonds between guanine and cytosine, and 2 between adenine and thymine, because this last one is just too far apart. It's more than 2 angstroms. So that's too far for an extra hydrogen bond. So that's actually an important detail, because later in the class you're going to, and in lab, you're going to talk about a technique called the polymerase chain reaction, where it's important to know the composition of your piece of DNA to know how much energy it's going to take to separate your strands of DNA. And separate the strands means separate this from this. So this is on one strand, this is on another strand. If your DNA is made up of a lot of these sort of base pairs, A with T, that's 2 bonds between those base pairs versus 3 bonds between these guys. And so if you have a lot of AT, it takes less high temperature to separate these 2 strands. And if you have a lot of G and C, it takes higher temperature to separate those strands because you've got to break more bonds if it's between G and C than if it's between A and T. And that's important in that PCR reaction that you'll talk about later on in class and in lab. Okay. C and T are called pyrimidines. That's the type of molecule that they are. That's the chemical name for them. G and A are called purines. That's the type of molecule that they are. How I remember this is A and G spell the word ag, and purine and C and T. There is another pyrimidine that we'll talk about when we talk about RNA. That's called uracil. That's another kind of base that's only in RNA. So uracil is a pyrimidine just like C and T. And C, U and T, U for uracil, make the word cut. So because there's 3 pyrimidines, and pyrimidines is a longer word, that's how I remember what's a pyrimidine and what's a purine. So pyrimidines, 3 bases are pyrimidines. Purines, 2 bases are purines. A and G are purines. C, U, and T are pyrimidines. Okay, A always binds with T, except sometimes if we're talking about DNA. Later on when we talk about RNA, we'll break that rule. But right now we're just talking about DNA. And A always binds with T, and C always binds with G. C always binds with G no matter what we're talking about, RNA or DNA. Okay, so A with T, C with G. A with T is 2 hydrogen bonds. C with G has 3 hydrogen bonds. It takes more energy to break a CG bond than it does an AT bond. And one thing that Watson and Crick noticed and laid claim to in their original paper was the idea that if you know what this strand is, you automatically know what that strand is. So if you know the sequence of this strand, let's say a G and an A, then you know on the other strand that G is binding with a C and that A is binding with a T. So if this strand is G and A, this strand is T and C. So if you know what one strand is, you also know what the other strand is. So I'll give you an example of that. Sometimes, in fact, most of the time, when we're talking about DNA sequence, we only write the bases down and the 1-letter code for the bases. So this is adenine, guanine, cytosine, thymine, so on. The sugar is on here and the phosphates are on here, but because those are the same for all the bases, we don't include those. We just write the bases. Okay? And here's the 5' end and here's the 3' end. So this is where it starts. That's nucleotide number 1. This is the last one. And if I was going to make this longer, I would have to add on to this end. I could not add on to that end. So I know what this strand is. What's the other strand? Well, it has to be this, because A binds with T, G binds with C, C binds with G, T binds with A, and so on. And this strand here is anti-parallel. Okay? So it's 5' to 3' left to right. This is 3' to 5' right to left. Okay? So anti-parallel and A with T, G with C. All right. So that was a big advance that Watson and Crick made in that the structure of DNA, and they won a Nobel Prize for it. And again, they didn't give any credit to Rosalind Franklin, who deserved to be on the Nobel Prize, but she died soon after their publication, and you can't win a Nobel Prize if you're dead. So the next big question was, okay, now we know what DNA looks like. How is DNA replicated? So scientists knew that you replicate your DNA before cells divide because your new cell needs DNA. And so you have a copy of your DNA in your old cell, you need a copy in your new cell. How is that DNA replicated? And there were three possibilities. And this is going over those three possibilities. And in this diagram, the darker colors here, like dark purple and dark teal here, those represents the original two strands, anti-parallel strand, 5 prime, 3 prime, 3 prime, 5 prime. Okay, and then these lighter colors here and here represent the strands that are made to replicate the DNA. So the lighter strands are the replicated strands, the darker strands are the original strands. So here are the three possibilities for how DNA replicated itself. One possibility is that the semi-conservative model, so that you have these two strands, they separate, and then each one makes a copy of itself. So dark purple makes light purple, dark teal makes light teal. And then you end up with what was one double-stranded piece of DNA becomes two double-stranded pieces of DNA, and you could replicate your DNA that way. That was one possibility. Another possibility is that each strand is just totally replicated by all new DNA. So it wasn't that the old strand is as a template to make a new strand, is just copied completely into new. And so that your new DNA was all new. Versus in this situation, this double-stranded piece of DNA was half old and half new. In this situation, your new DNA is all new. And this is called conservative replication. And then there was another possibility in that you wouldn't necessarily replicate a whole strand at once, but maybe only part of a strand. So you have a part of a strand of DNA, and then you make a new strand within that strand of DNA, and then you go back to the old strand, and then some new strand. So each of the strands was a mosaic of old and new. Each of the new strands was part old and part new within a strand. Versus in this situation, this has one complete strand that is old and one complete strand that is new. In this situation, each of the strands is part old and part new. And this is called the mosaic model. So there's some scientists called Meselson and Stahl, and they designed an experiment to find out which one of these possibilities is the real thing. How does DNA actually work to replicate itself? Okay. What they used was radioactively labeled DNA. Radioactively labeled DNA that was labeled with nitrogen that had 14 neutrons or 15 neutrons. 14 N14 was called light, and 15 was called heavy because it had a larger mass. And only one neutron difference is going to be really hard to measure. There's no scale that could measure that sort of thing. And so what they did was they had lots and lots of DNA, many millions of copies of the molecule that were labeled either with nitrogen that had a molecular weight of 14 or nitrogen that had a molecular weight of 50. Okay? Then what they could do is they could put their DNA in a centrifuge tube that had a gradient of salt. So not so salty at the top and very, very, very salty at the bottom. And a continuous gradient all the way through. And salt, if you've ever been to the Great Salt Lake in Utah, or if you've seen pictures of it, you can look up on YouTube. Salt acts to like a buoyancy to hold things up. So the DNA molecule is going to have a certain density, and at a certain density of salt in this gradient of salt, it's going to float, and it will not sink anymore. Even if you use the centrifuge to try to push it to the bottom of the tube, it won't do that. And so DNA that started off here and is actually more buoyant than that. The salt will push it back up. To the density where it matches the density of the salt. And if it's heavier than the salt that it's sitting in, it'll sink until it finds that magic density where it matches whatever the density is of the salt. And in this experiment here, when they put it in a powerful centrifuge and they spin it for a very long time, the force of the centrifuge will push up DNA that is more buoyant than the salt. The salt will push it up, and if it's less buoyant than the salt, it'll sink down until you get a single band after centrifuging, and that's where that concentrates all of that DNA. And if the DNA weighs a different amount, it'll have different buoyancy in the salt gradient, and so you can separate DNA that is labeled with nitrogen 14 versus nitrogen 15. That's the idea. Okay, and in fact, and then they could see the DNA in the centrifuge tube with ultraviolet light because DNA shines bright in ultraviolet light. So they get a band of DNA that is labeled with 14 nitrogen at one spot, and 15 nitrogen shows up at a different spot, okay? And there's some distance in between them. So they can tell the difference between DNA labeled with 14 and DNA labeled with 15. And they can actually quantify how much DNA is at each spot by taking a picture of the DNA in the centrifuge tube, and then they just count how dark is it here, how dark is it here, how dark is it here, and then it starts to get darker and darker, darker. That's going up the peak here, and then it starts to get lighter and lighter and lighter, coming down the peak there, and then goes back to light in between, and they plot that as they go from the top of the tube to the bottom of the tube. They get these two peaks, and then they take the area of the peak, and that quantifies how much is at the 14 spot, how much is in the 15 spot. This technique here is used a lot in molecular biology, and trying to quantify something, a picture of something, like on a technique called Western blotting, for example. Okay, so here's how they did this experiment. The scientists are called Meselson and Stahl. In there, they did two experiments that are graphed here. In the first experiment, what they did was they measured the density of cells, bacterial cells, that bacteria have DNA too, and they labeled those bacterial cells and they sampled them every once in a while, and then they measured them over a certain amount of time. So first, they supplied the bacterial cells with heavy nitrogen, N15, and at a certain time, they switched to the only nitrogen being available is light nitrogen. So if these bacterial cells are replicating, okay, there's more and more cells in the experiment. At a certain spot, they said, okay, you've been using heavy nitrogen, now we're going to switch to light nitrogen, and we're going to look at what your DNA looks like. So these ancestor cells would be made up of DNA with heavy nitrogen. When we switch to light nitrogen, what does the DNA look like? They also did the experiment a second time, okay? And they had DNA cells at a less density. That's why the line is lower. But the same idea. First, heavy nitrogen. Second, light nitrogen. Okay. First, all cells were grown in heavy nitrogen. And then they harvested those cells at time 0, switched over to light nitrogen, and then harvested these cells at a bunch of times and say, okay, when do you start to see light nitrogen showing up in your DNA, and what does the DNA look like? Okay, so they take the DNA that they sampled at these different spots, they separate it in the salt gradient. Again, they should be able to tell the difference between heavy and light, see what happens. Okay, so when they look at the part of the experiments where all they got was heavy nitrogen, they get a band in the density that indicates that all the DNA that they've got is heavy nitrogen. It shows up lower in the tube and it's all heavy. Okay, that makes sense. That's all they had to work with. Okay. Then when they switch to all light, first it looks like it's in between, and then later, oops, it looks like there is two bands. One of the bands that is all light, all N14, and one that is in between. So it's not like you have a band here and a band over here, not all light and all heavy. You have some that is all light and some that is partially light and partially heavy. Okay, so what does that mean for the 3 models of replication? Okay, so this band means that it's the 2 strands of DNA are 50-50, 50 light, 50% light, 50% heavy, and then some of the DNA is 100% light. All right, so let's look at these models and figure out which one could that be. All right, so all heavy is what you started with, and here's the results where you got something in between. You don't end up with all light. So if conservative replication was the way to go, you take your old DNA and you make totally new DNA from that, which didn't have any old DNA in it. And that's not what you saw. So here's the old DNA, and the new DNA should be over here, not someplace in the middle. So this conservative replication can't be it. Okay? What they saw was 50% of the DNA was light and 50% was heavy. So, and then the new strand of DNA, whereas 100% was all light, that knocked out this as a possibility. Okay, so let's go back here for a second. All right, this where it's half light and half heavy, this could be either of those two models. It could be the mosaic model, or it could be semi-conservative model. But as the experiment kept on going and cells are replicating more, now in the second generation of cells, they're using old DNA, which is now light because they've gone for several generations when all they had was light. And the new DNA they make from that is also light. So they end up with 2 strands of light. Okay? So they had, in the first replication, they had 1 strand of heavy, 1 strand of light. And then the next time they have 2 strands of light. Okay? If it were mosaic, then always the case would be that you'd have this band in the middle. All the new DNA you made would be half old and half new, no matter how many generations you did it, if this is the way that DNA replicates. And because they saw this band here, they know that can't be the case. Okay? So, this can't be true because they saw They didn't see just light DNA after they switched to N14. This can't be true because they didn't see just 50% light and 50% heavy in the new DNA. This has to, this has to be true in that in the first generation, the first time they get light DNA, they get DNA that is half heavy, half light, half heavy, half light. Now when this new DNA, the light stuff here, replicates again, it's going to replicate using light N14 again, and you'll get 2 strands of light DNA as a result of that, and that's how you got this band here. Okay? So, you get this band from the first time the DNA replicates. You get this band the second time the DNA replicates. If this was the model here, you would get this band forever, and this band would never show up. Okay? So, this is the way that DNA is replicated. You have an original strand that is double-stranded. It separates into 2 strands, and one strand here, the darker strand, acts like a template to make the new strand. This strand here acts like a template to make the new strand. In their experiment, the template was heavy nitrogen, the new strand was light nitrogen, and so that's how they got a band in the middle. So what do I mean by template? Let me go back here. So this is what I mean as a template. If this is your original stranded DNA, and you separate from 2 strands into 1 strand, then the machinery that makes DNA, and we'll talk about this later on in the class, DNA polymerase, it will use this as a template and say, okay, I've got an A right here. What do I put on the new strand? And here's the new strand. If there's an A here, I must put a T here. If there's a G here, I must put a C here. If there's a C here, I must put a G here. And so the new strand of DNA is half old and half new. That's called semi-conservative replication.